Hey Geeks, welcome to another video. So today we will be discussing all you need to know about antibodies for any of your entrance examinations. So in this video, we will be discussing about the structure of the different antibodies, the classes, the subclasses, the abundance, and the function of each of this. Along with this, I will also be describing terms such as idiotype, allotype, isotype. So please stick to the end of the video to get all of these answers. And thank you so much guys for liking, subscribing and commenting on all my videos. I really appreciate all your support. Without much further ado, let's get right into it. So firstly, I want to tell you about the serum electrophoresis. So basically when you put serum in presence of two electrodes, that is, you let the particles migrate under the influence of the electric field, we observe such a pattern. So this is the origin, and from here is the negative terminal, the positive terminal. What we observe is you get firstly a band of gamma globulin followed by beta globulin, beta 1 globulin, alpha 2 globulin, alpha 1 globulin, and the majority portion is of albumin, which is found at the positive electrode. Okay, so remember this. There are questions that are asked that which is the majority protein present, where will be the band. Okay, so remember this. Next is about the antibodies. So basically, antibodies constitute 20% of the total proteins in the blood plasma. Antibodies are heterodimers, that is, they consist of two light chains and two heavy chains. The two light chain can be the kappa or the lambda. Both of these are seen in humans. However, it can either be both the light chains can be kappa or both the light chains can be lambda. They can never be one kappa, one lambda. Next is the heavy chains. So basically, the heavy chains are the basis on which there are different classes of antibodies. So basically, there are 5 different heavy chains. That is, first one is the gamma chain. So those antibodies that have the gamma heavy chain are said to be the IgG, the mu chain IgM, alpha chain IgA, delta chain IgD, and epsilon chain IgE. So these are the 5 different classes. Remember, henceforth, if they ask you a question, the classes of antibodies are based on, yes, the different heavy chains. Now, there are some facts that I want you to know. So firstly, the N terminus of these antibody molecules are variable. However, the C terminus is constant. The antigen binding sites are comprised of the N terminus of the light and the heavy chains. The variable region contains two major divisions. One is called as the hypervariable region, which is further divided into other divisions, and the framework region. So usually the framework region remains constant, but there is a change in the hypervariable regions. So there are 3 different hypervariable regions called as the CDRs. CDR stands for complementarity determining regions, and they are 3, that is CDR1, CDR2 and CDR3. CDR3, remember, is the most variable. The light chain is composed of one variable and one constant. The heavy chain is composed of one variable region plus either 3 or 4 constant regions. So for the antibodies IgM and IgG, you have one variable region and 4 constant regions. The rest of them have only 3. That is the IgG, IgD and IgA. So IgG, IgD and IgA have one variable, 3 constant regions. Next, there is a presence of the hinge region in basically the IgG, IgD and IgA antibodies. The heavy chains of these have slight different genes, because of which they basically form this hinge region, and the hinge region is found to be rich in proline and cysteine. So the heavy chains of these IgG, IgD and IgA, they have what is called as the CH1 and CH2 regions that are not present in the rest. The heavy chains have the hinge region, they make the hinge region which have proline and cysteine, which are basically known to give flexibility to these antibodies. Okay. Now, this is the typical structure of an antibody. Now, the primary structure of the antibody is composed of straight chain amino acids, that is for the variable and the constant regions of the heavy and light chain. The secondary structure basically consists of anti-parallel beta pleated sheets. The tertiary structure is consisting of compact globular domain, and finally quaternary structure of the antibody is when the adjacent heavy and the light chains interact to form the different domains. Okay, so primary structure, straight chain of amino acids. Secondary, it is basically the beta pleated sheets in the anti-parallel arrangement. Tertiary structure is when the globular domains are found, and finally the final structure is when the adjacent light and heavy chains interact. Now, immunoglobulins are of two types. One is secreted and second is membrane bound. So as the word suggests, secreted, so they are given outside, and there must be some difference between these two immunoglobulins. The difference is that the secreted ones have hydrophilic amino acids at the C terminus, whereas the membrane bound, they have three different regions: the extracellular hydrophilic spacer sequence, the hydrophobic transmembrane sequence, and finally the short cytoplasmic tail. Now, coming to the different classes of the antibodies. So firstly, we have the IgG. Now, IgG is the most abundant, that is, it forms 80% of the total plasma. It has four subclasses, that is IgG1, IgG2, IgG3, and IgG4. The numbers are given according to the decreasing order of their concentrations in the plasma. The half-life of IgG is 23 days, which is actually the maximum, and except for IgG3. IgG3 has a half-life of only 7 days. It is the only antibody that can pass through the placenta except IgG2. So IgG2 cannot pass through the placenta. However, the other three can. So this is a very funny meme and a very nice way to remember. So you will see that these different types of diagrams will be there further. It will help you to remember. This credit for the meme goes to the page Biomimics. I will link down their Instagram page. You can go follow them. The next one is IgM. So now IgM is constituting 5 to 10% of the total plasma proteins. The half-life here is 5 days. Now, IgM is always first. How? First antibody that is made by the B cell. It is also said to be the first antibody made by the neonates. Okay, and during an invasion by a pathogen, it is again the first antibody that is made, after which by class switching IgG is produced. It is also more efficient in activating the complement system than your IgG antibodies. And what is the reason? Because for activating complement, you need two FC regions, okay, two framework regions in close proximity. Therefore, this is basically found to be more efficient. Next is the structure. So IgM, if it is membrane bound, it is monomeric. It is, if it is secreted, then basically it is present as a pentamer. So this is a very nice way to remember a pentamer. It is having two heavy chains, two light chains. These are joined together, all these units are joined together by disulfide bonds between the FC regions, and they also contains something called as the J chain. So the J chain is basically a polypeptide that joins this. This is said to be basically pentavalent because it can bind now. It has 5 valencies, so it can bind to 10 different antigens. Okay. Next, IgA. So IgA is present as 10 to 15% of the total plasma proteins. It has a half-life of 5.5 days and has 2 subclasses, that is IgA1 and IgA2. It is very predominant in all secretions, namely the breast milk, that is the colostrum, tears, saliva, and the mucus of the GI tract, urinary tract. It has a maximum daily production. The structure of IgA. So in the serum, IgA is monomeric, whereas in the external secretions, they are present as dimers or as tetramers. Again, the dimer and the tetramer contain a G chain along with another additional polypeptide called as the secretory component. Next is IgE. Basically, IgE has a half-life of 2.8 days, and it is mainly seen in the hypersensitivity reactions like those in hay fever, hives, anaphylactic shocks. These basically function by binding to the Fc receptors of basophils and mast cells that cause the degranulation of the prostaglandins that are present, and that lead to the allergic manifestations. It is a very important defense mechanism against parasites. Nextly, IgG. It is the least present, that is only 0.2%, and its biological function is actually not known. The half-life is only for 2 days. Now, it is basically present on the surface of the B cell. So along with along with IgM, the IgD is present on the surface of B cell that forms a complex called as the BCR, antigen specific B cell receptor. Okay, so this is about the IgD. What are the function of antibodies? So the basic function firstly is opsonization, that is they coat the pathogen so as it becomes more visible to the immune system and it attracts macrophages to come and destroy it. Next, toxin or viral neutralization. So these antibodies have the capability of binding to viruses that have not infected the cells yet, and they can prevent their further infection and entry into the cell. Activation of complement, again it works in the same manner as opsonization. Immune complex formation, ADCC. What is ADCC? Basically, it is antibody dependent cell cytotoxicity. So basically, antibodies, they coat the pathogens and they are then leading to attraction of the other phagocytic cells that come and engulf the bacteria. The immune complex formation is the antigen is neutralized by the antibody. They form a complex and now the antigen is totally destroyed. This complex is basically come and engulfed by the other phagocytic bacteria and they are destroyed. The immune complex formation results into 2 types of reactions, that is aggregation and precipitation. Now, since antibodies themselves are glycoproteins, they can function as immunogens. So basically, the epitopes or the antigen determinants of the antibodies, they fall into 3 different categories. First category is called as the isotype. Now, what is the isotype? So look at this diagram and try to understand what I'm saying. So basically in humans, as we discussed, there are 5 isotypes, that is the 5 different classes of antibodies. Now these 5 different classes are basically based on what? The heavy chain, right? So what is the heavy chain? That is nothing but this constant region that is different in each of the immunoglobulin. So isotype mein, each isotype corresponds to separate constant regions. Okay. Now all members of a species carry the same constant regions. So IgG, IgA, they will all be the same in everybody's body. Next is allotype. So in allotype, this is due to the genetic differences in individuals. It is basically related to the concept of alleles. So one gene can have many allelic forms. Similarly, each human may have different alleles for this IgG. So although they both are human IgG, there are slight differences in the amino acid sequences. This is allotype. Again, like the previous one, like the isotype that we studied, it is also a phenomenon that is found because of the constant region, seen in the constant region. Next is idiotype. So in idiotype, as you can observe, the variable regions are involved. So the variable domains of the antibodies function in two aspects. One, they act as antigen binding sites, and two, they also have these antigenic determinants. Now, how are these determinants created? So basically, with the different conformations of the heavy and variable chains, these basic differences are formed. So two IgGs can be completely different in the same individual because of the variation in these variable regions. So this is called as idiotype. Also, there is one section that I have not included here, that is the on digestion with the different enzymes. So basically, three scientists, Porter and Endelman, used papain enzyme. Then Endelman himself used mercaptoethanol, and Nissenov used pepsin. And there were different fragments that were formed. So if you want this information, maybe I can put it in the comment section somewhere for you, or else I'll just leave it here. So that's it from me for today's video. I hope you enjoyed the video, you learned something new. If so, please like the video and subscribe to my channel. I'll see you in my next one. Bye.